Hi guys, my name is Lexi Garner and today I'm going to be going over the highlights of my internship this past semester. So for the past 4 months, I've been interning in the Pollinator Conservation and Ecology Lab with Rachel Mallinger, and I've been working with one of the PhD students named Sarah Anderson on one of her projects. My first project that I was assigned to had to do with slide observation methods. So basically I was given 8 slides, and the goal of this little experiment was to determine the most efficient way of observing pollen slides under a microscope in order to observe the greatest pollen diversity. So the 4 different methods were starting in the upper left-hand corner and going down and up until I get to either 100 pollen grains or to the end of the slide. The second method was starting in the middle top and doing that same process. The third method was starting from the left middle and carrying out that same process. And then the last method was just to count pollen grains in the upper right-hand corner. So the results from that experiment, we determined that the greatest pollen diversity came by doing the first method, which was starting in the upper left-hand corner and then just going through until we got to the end of whatever, either the 100 pollen grains or the end of the slide. So that was my first couple weeks at the lab doing that experiment. And then I was assigned to the actual project that Sarah and I have been working on for the last 3 months. So the project's goal is essentially to answer whether or not southeastern blueberry bees' foraging habits or foraging preferences change over across habitat types. So the southeastern blueberry bee is supposed to be a specialist on Florida blueberry bush, but what we're seeing is that when they are in disturbed areas, they are kind of going to other sources. They're not necessarily just going to blueberry bushes, and so that was the data that we're trying to collect. So in the spring, Sarah collected southeastern blueberry bees from urban sites, natural sites, and agricultural sites. After that, we took the pollen, we scraped it from the body and the legs from the bees, and we put them onto pollen slides. My job for the past 3 months has been to go over each and every one of those pollen slides and record the pollen diversity. So we are essentially seeing whether or not the southeastern blueberry bees are foraging on just blueberry bushes or if they're foraging on a diversity of different plants, especially in urban habitats where there may not be as many blueberry bush as usual because they're disturbed areas. We're actually completing the data this weekend, so I don't really have any results to report just yet, but we are seeing a trend that the southeastern blueberry bee has no preference between other plants. So they're basically foraging on the same amount of blueberry plants in agricultural sites as they are in native sites. So basically there are 2 different types of bush, species of blueberry. The agricultural site is obviously a certain type for agricultural purposes, and then there's the native Florida blueberry bush, and we're seeing that they don't really care which one they're foraging on. They're just foraging on whatever is available to them. So let's see what else. Some of the highlights besides that, I can't wait to see the data that we come up with this week and present that in my final presentation for you guys. But I do have a lab presentation coming up on Monday that I am very excited for. So basically I'm going to be presenting on my work experience the past 4 months to the entire lab, and then getting some feedback on the project that I've been working on. That's something that the lab does every single Monday, so it's really cool to get that kind of feedback. Each person in the lab presents on a different Monday for whatever they've been doing, whatever project they've been working on, and then the lab members give feedback, which is really, really nice, especially since COVID is keeping us apart and we have no way to conversate or converse on projects together in the lab. So it's really nice that we do that every Monday. And yeah, so that's pretty much a brief summary and the brief highlights of my work experience. And let's see, I would have to say that my advice for future plant science students would be to just go in with an open mind. Don't shut out in a work experience just because it's not directly in your field. I obviously am a plant science major and I'm working in the entomology lab. I have been working in the entomology department for the past 2 semesters. I love it. I worked in the honeybee lab last semester, and I worked obviously in the pollinator conservation and ecology lab this semester, and I love it. I would also say that make relations. I know that in the honeybee lab, I didn't really get to connect with people all that much. And this time around, I've been able to connect to my lab members a lot more through the lab meetings and working one-on-one with Sarah every single time that I come into the lab. It's been really great. So I would just say to make connections so that you have references for the future, potential job opportunities. I was actually offered a job in my lab for next semester, so I'll be doing field research with Sarah for the same project in the spring. I'll be going out to a bunch of different sites all over Florida, collecting southeastern blueberry bee and recording all of the vegetation and plants in the area. So I would just say, go out of your way to make those connections, because it's only going to help you in the future. So thank you guys so much for listening to this, and I will leave some pictures in the discussion board for you guys to see some of my different pollen morphotypes.